Hi everyone, welcome to my channel. In this video, we'll be talking about homeostasis. This is the most important topic of general physiology. Okay, in this video, we'll be talking about different types of homeostasis. Not only we'll understand this concept, and also we will see how to write in examination. If you want Hindi video, you can visit my main channel Physiology Hacks. I'll drop the link in description box and you can have a look. Without further ado, let's get started. Homeostasis. Homeostasis. Homeo means same. Stasis means which is not changing, right? So homeostasis, what's that? Basically, it is the maintenance of stable and relatively constant environment. Here, environment means our internal environment. What that mean? Basically, there is a concept of milieu interior given by Claude Bernard. Claude Bernard said he gave the concept of milieu interior and saying that extracellular fluid is our internal environment, which is very important for us. Okay, so he coined the term milieu interior. This extracellular fluid he called blood as extracellular fluid, and that composition is very important for us. He just stated this as milieu interior, right? Then homeostasis term is coined by Walter B. Cannon. Okay, he explained that to maintain this ECF, extracellular fluid, which is blood, to maintain the composition of this blood, the extracellular fluid, various feedback mechanisms are working. Okay, and he gave down homeostasis for that, right? So for us, what is important? We have to understand what is homeostasis, what are different parts of homeostasis and types of homeostasis. So basically, what is homeostasis? Means the extracellular fluid have some changes, and those changes have those changes should be brought back to the normal one, right? So for example, if a concentration of any substance is increased, so there are certain mechanisms which will work to bring it to the normal value, right? So those are homeostasis. Now there should be some signal, there should be some sensors which can sense that change of the signal, right? And then there should be some mechanisms which can control it, right? So that's why we have some components of homeostasis. The first component is sensor or receptors, right? For example, there is increase in blood pressure, which is sensed by some receptors present in the blood vessels. Those are baroreceptors. Okay, so baroreceptors, they sense the change in BP as high BP, and they send this signal via some nerves to medulla in brain, right? Then medulla is the coordinating center or control system, which is the second component of homeostasis. It do some activity and it stimulate respective neurons to be stimulated to do some action. For example, if there is increase in BP, what should be done? There should be something done to decrease the BP, like for example relaxation of the vessels. Constriction of the vessels increases the BP. So if the blood vessels relax, they will reduce the BP. So the components medulla will stimulate to the components, for example, vagus nerve. So parasympathetic component is going to relax the blood vessel. So medulla is going to stimulate vagus nerve to cause vasodilation. And vagus nerve is also causing decrease in heart rate. So both the things ultimately decrease BP, right? So the basic idea I want to give it here, that component of homeostasis just include some stimulus, then receptors or sensor, some control system and effector, and ultimately they all together will reverse the change. Fine, that is homeostasis. Now, this is just a simple idea. What we are going to explain if long question is being asked in homeostasis. So, all the mechanisms which play role in homeostasis, we call them feedback mechanisms. Okay. So, we have three types of feedback mechanisms. One is negative feedback mechanism, other is positive feedback mechanism, and there also one exists which is feed forward mechanisms. Right. The most important among this is negative feedback mechanism, which 90% of our body change, any change in our extracellular fluid, that is blood here, is basically managed by negative feedback mechanisms, right? Okay, so let's see what you are going to explain in these three mechanisms. So first is negative feedback mechanism, right? What is negative feedback mechanism? Basically, whenever there is change in one parameter, for example, this parameter we are talking about temperature, right? So if there is increase in temperature, right? What happens basically that high temperature, there should be some sensors present to sense this high temperature, and here the receptors are skin or brain which can sense the high temperature, right? Then they send some signal to hypothalamus, which is control system for temperature, and it sends some signal to skin or vessels. What they do, if there is high temperature, that can be reduced by sweating or vasodilation, right? So increase in sweating occur via effectors on skin, and vasodilation occur via effect on vessels. Because of this, there is increase in heat loss, and obviously when there is increase in heat loss, the temperature can be brought back to normal. Okay, so there was a change, there was a sensor, coordinating center, and effector. But what is negative feedback here? Negative feedback mechanism basically respond in the reverse direction of the change always. When the change is increased and effector do the vice versa, the opposite of the change, the decrease in temperature. So basically the stimulus, the response is opposite to the stimulus. If this happens, this is negative feedback. So ultimately, whatever we are managing, it is negative to the change happen, opposite to the change happen. Let us see one more example to understand negative feedback mechanism. Okay, so example 2 is blood pressure. But even in temperature, if there is reduction in temperature, decrease in temperature, there are some mechanisms which stimulate shivering via hypothalamus only. They stimulate shivering and in workout basically, which increases the heat production and increase temperature. So this is again negative feedback mechanism. The response is opposite to the change happen. Second example is blood pressure. As we have just saw that this increase in blood pressure sensed by baroreceptors. Baroreceptors are the sensors which sense this high temperature, high blood pressure. Okay, then they send this signal to medulla. They stimulate the parasympathetic system, which can reduce it, and inhibit the parasympathetic system, which can increase it. So ultimate result is vasodilation and decrease in heart rate, which is going to reduce BP. Now, if you don't want to repeat this example here, you have many examples present over there in our body. All the hormones, approximately all the hormones work as negative feedback mechanism. So whenever, for example, thyroid hormone. So pituitary is going to secrete TSH, thyroid stimulating hormone, and which acts on thyroid gland to stimulate, to basically secrete thyroid. So in response to TSH, thyroid gland secrete thyroid hormone, right? So thyroid hormone, whenever there is high concentration of thyroid hormone, it is going to negative feedback. It is going to give negative feedback to pituitary to inhibit the secretion of TSH hormone. So ultimately TSH will be reduced, which will not stimulate thyroid gland then. So ultimately thyroid is reduced. So increase in thyroid is going to influence decrease in thyroid concentration. Again, here this is negative feedback. So approximately all the hormones work in the same way as negative feedback. They basically regulate their concentration via negative feedback mechanism. Okay. Now let us see what is positive feedback mechanism. Now I hope you can make this definition that the response is in same direction. If change is happening in one direction, you are going to stimulate, some mechanisms are going to stimulate in same direction of stimulus, right? So what are examples for this? There are various examples although, for example, parturition reflex. So whenever labor occurs, so head of baby, when it's fixed in cervix, it stretches the cervix, right? During that point, it stretches the cervix. Whenever cervix is stretched, it basically releases oxytocin. It basically stimulates pituitary to release oxytocin. And whenever oxytocin is released, it basically causes uterine contraction. Whenever uterine contraction occurs, there is cervix below obviously, which will be stretched again. So uterine contraction are going to stimulate and stretch the cervix to stimulate to basically stimulate pituitary to secrete oxytocin, posterior pituitary, right? And more oxytocin means more uterine contraction, means more stretching of cervix, means more uterine contraction. So ultimately, this is a vicious cycle which is going to stimulate each other. And that's why we call that if the change happened, whatever change it is basically stimulating it via various mechanisms. And those mechanisms are positive feedback mechanisms. There are various other examples as well. For example, example 2 is blood clotting. We have cascade of reactions activating the blood clotting factors. If you can see, if you remember, ultimately there are various factors. For example, if we talk about intrinsic pathway and extrinsic pathway, there are various factors, series of factors which activate each other. And ultimately factor 10 is formed, which is prothrombin activator. Now factor 10 is going to stimulate the thrombin formation, and thrombin formation is going to stimulate formation of factor 10. Okay, thrombin which stimulate more factor 10 formation. So ultimately thrombin is formed, it help in fibrin formation at last. So they are basically activating each other. That's why it is positive feedback mechanism. There is one more example like milk ejection reflex. Suckling of baby, it stimulates pituitary gland to secrete oxytocin, and oxytocin is going to stimulate milk ejection. Whenever milk ejection occurs, baby suck more. Baby suck more, that means it again stimulates pituitary. So again this is a vicious cycle which is a type of positive feedback mechanism. There is nerve impulse generation. The action potential generation is also one of the examples. More positive cationic influx in the cell is going to stimulate voltage gated channel more to open, which is causing the positive feedback for the action potential. Last is feed forward mechanism, which is basically now like active this positive and negative feed mechanisms. The change has happened, and now there are some mechanisms to reverse the change or do some response in the same direction, right? But here the change is not yet happened. Change has not happened here. Okay, there is no change at all, but we are just anticipating that change. We are just predicting there is some change going to happen. Okay, but just anticipation is going to stimulate some mechanisms to take action. For example, there is increase in heart rate before starting the exercise. If you are at start line, you haven't done any exercise, but your heart rate is high. That mechanism, that sympathetic stimulation is because of the anticipation that you are going to run, you are going to do or start the exercise, right? Second example is acid secretion before eating the food. Even when you smell the food, when you see the food, and when you have plate ready, right? Even seeing that food is going to stimulate the acid secretion, saliva secretion in our body. When you take the food in mouth, that food is not in stomach, but it stimulates acid secretion. So that is also example of feed forward mechanism, in which you just anticipate or you just predict the change to be happen, and just before that you take action, right? There is no gain or error correction happen because change has not happened. Okay, so it's just the preparation for any change. Now what is this gain or error? So gain of a system is basically telling you how much effective the system, the feedback system is. Gain of a system is basically correction by error. So gain of negative feedback, for example, is the degree of effectiveness of the feedback mechanism. How much effective it is, right? So more correction it is, more gain it is, and less error. Now we can understand by taking an example. For example, our BP increases from 100 to 150, right? Thus basically system tries to brought it back to 110. Okay, now he has basically brought back this BP at 110, not 100. Okay, it has increased to 150, but the system brought it to 110 only, not 100. Right. So what was the correction? Correction was 150 minus 110. So 40 mmHg is the correction. What is error? The final outcome is basically having 10 error. It should be 100 otherwise, but it's 110 only. So error is only 10 mmHg. So what is gain of the system? It's correction by error. So 4 is the gain of the system. Okay, this way we can see this feedback mechanism, how much effective it is. So that's all about homeostasis. I hope you liked it. We have completed these three mechanisms. I have shown you 3 to 4 examples, but if this question is asked in your examination, you can perform based on the criteria. If it's 10 mark, you can give 2 example each, and that will be sufficient for you. So I hope this topic is crystal clear to you. If you have any query, you can comment below. Thanks for watching, guys. Bye bye.